Hello, my name's Liam Friar. I'm a soon-to-be sixth-year PhD candidate at the University of Colorado in Boulder in the U.S. And I'm going to be sharing with you today some genomic insights from Trichomonas azola, which is an endosymbiont of this fern azola that's pictured here, grown in a greenhouse. And I'm going to be asking the question, could an extracellular endosymbiont become an organelle? And if that question sounds like nonsense, hopefully it won't in the next couple slides. So why study endosymbiosis and what does this have to do with organelles? We're pictured here mitochondria and the chloroplast, the famous powerhouse of the cell and photosynthetic organelle of eukaryotes that both represent major evolutionary milestones and are both widely accepted to be descended from ancient bacterial endosymbionts. And it would be awesome to understand how you go from a free-living bacteria to an organelle that's super highly integrated with its host, such that we just call them one organism. But unfortunately, studying these organellar events directly is difficult since they're so ancient. So instead, we can study modern proxies. And I would argue that cyanobacteria are a really ideal system to do this in, because they sit all along a spectrum of integration with the host. If we start in the top left here and go clockwise, kind of integration increases. So from a free-living bacteria to a facultative endosymbiont that can be found with or without its host, to an obligate endosymbiont of varying degrees of integration, to endosymbionts so integrated that we call them organelles, and then obviously the chloroplast, this ancient super integrated organelle. And in all of these instances, the cyanobacteria is either supplying its host with carbon that it fixes or with nitrogen that it fixes. And so I'm talking just about one of these systems, the host plant being fern azola. So azola is a genus of 4, give or take, depending on who you ask, species of ferns, so a land plant. But as you can see on the left, or on the right, they grow on the surfaces of fresh water, the far left being a bird's eye view of me growing some in a greenhouse again. As you can see from the scale bars, they're quite small and they grow extremely fast. They can double their biomass in as little as 2 days. This isn't what I'll be talking about, but is kind of a really interesting thing about them. There's a hypothesis that seasonal azola blooms about 50 million years ago may have been drawing down so much carbon dioxide from the atmosphere as to affect global climate, because they grow so fast. They have a really broad geographic distribution, and the azola genome was the first complete fern genome published. So this is a bunch of cool stuff, but it was what is like the coolest to me about this system. What I'll be talking about is the symbiosis that it hosts. So this diagram taken from Carapace. If you look on the far left is a cross-section of a leaf. So inside of each of these leaves, there's this fully enclosed cavity filled with mucilage where a population of these filamentous cyanobacteria live. You can see one of the filaments there in the center. So some people don't like the use of the term endosymbiosis because it's extracellular. I'm going to still use the term, but regardless, it is obviously different from these other endosymbioses that I showed, and certainly from the chloroplast and mitochondria, in that the symbiont is fully enclosed within the host, but it is extracellular. So is it still going to evolve in the same way, or is that going to make a big difference in its trajectory? So a little more background. The symbiont is called Trichomonas azola, shortened to TAZ. It's a member of the Nosticales, which is an order of cyanobacteria. They're filamentous and differentiate different cell types and morphologies. One example being heterocysts, which are the nitrogen-fixing cells. You might see one of the cells in that filament is much larger and more like an oval, so that's where nitrogen's fixed, whereas photosynthesis happens in those other cells. Several of the Nosticales form facultative symbioses with a wide range of plants. And in all these cases, the Nosticales is fixing nitrogen, so taking dinitrogen from the atmosphere, turning it into ammonium, which the plant can use. And that's exactly what's happening in this symbiosis in azola. But what's unique about this one is that it's an obligate symbiosis. So neither partner is found in nature without the other. And the symbiont is passed from mother to daughter plant, passed vertically. Yeah, and there's also a reference genome published for the endosymbiont. Now this image taken from the literature is autofluorescence of the symbiont, just to give you a better idea of what things look like. So again, on the bottom in isolation, you can see the symbiont has those different cell types, and on top, you can see it fluorescing through the leaf pocket. So there's a single origin of this symbiosis, a single symbiotic event, 50 to 90 million years ago. The host can grow in isolation, although it's never found like that in nature. If you treat it with antibiotics and then give it a bunch of nitrogen, it grows totally happily on its own in lab. But the symbiont can only survive for maybe a week or two in isolation, and nobody's ever gotten it to multiply or really culture it. The symbiont does perform photosynthesis when it's in isolation. However, it's still somewhat undetermined the degree to which it's doing this when it's in symbio. And along with that, it's not really clear. A lot of things about what's happening when the symbiosis is intact aren't really clear. Are they both fixing carbon? The plant certainly is, but is the symbiont? Are they exchanging carbon? Are they exchanging anything else? So basically, the degree of integration between the partners is still unclear. All right, so this is a genomics talk. So endosymbiont genomes evolve in characteristic ways. There's a bunch we could talk about, but 3 in particular that are all related. Genes break, they become what are called pseudogenes. Genes disappear completely, and there's weak, or if you prefer, inefficient selection on many of the remaining genes. And since this is a short talk, we're just going to focus on that last one, weak selection. So forgetting about all the broken genes. And again, we're talking just about the genome of the endosymbiont, just to make that clear from the get-go. We're not talking about the host at all in this talk. So kind of 2 broad umbrellas for reasons that you might see weak or inefficient selection. One, some genes are maybe just not as important in the host environment. For instance, maybe you never have to deal with high salt, you never have to deal with heat, et cetera. So those genes are, there's not a strong purifying selection for them to still work well. Alternatively, population dynamics characteristic of an endosymbiont lifestyle, such as frequent bottlenecks, lack of diversity, lack of recombination, can lead to kind of drift or very weak selection even on genes that are still important to the symbiosis. So we can picture kind of different outcomes that we might expect depending on these 2 possible causes. Here you've got a diagram of a bacterial chromosome in gray. Different genes are the different colors around the circle, and we imagine that we can color them from least to most important to the fitness of the symbiosis, with least being yellow, most in red. And then we imagine we can figure out where there is weak selection, and if the first hypothesis that it's about some genes just not being important anymore is true, then we put black X's, the weak selection, just on those genes that aren't that important. But if it's those population dynamics and it's kind of more of just drift happening, we might expect that there's weak selection on a wider range of genes and also just more widespread. It's not just limited to those genes that aren't important. All right, but is this what happens in an extracellular endosymbiont? So is there evidence of weak or inefficient selection in the Trichomonas azola genome? If so, what appears to be causing it? Which cellular functions are most and least affected? Those are the questions we're going to try to answer. And to do that, I compare genomes from 8 Trichomonas azola strains and 40 free-living Nosticales, so the closest free-living relatives, all taken from public repositories. And again, this is the symbiont and cyanobacteria, not the host, not the plant. First look at the data. We're able to build a phylogenomic tree. The Trichomonas azola genomes highlighted with the green bar and blown up in the inset are monophyletic. That's good because there's a single symbiotic event. And those dates are taken from the literature. And we also see that or just to tell you, we don't see it here, the phylogeny of the Trichomonas azola matches previously published phylogeny, so that's a good check on our data. And it perfectly matches the host phylogeny, which was one of the first pieces of evidence that this was a vertically passed single origin symbiosis. But now the meat of what we're getting to, strength of selection. So without getting into the weeds, DNDS is a ratio of 2 types of point mutations. Just very briefly, S stands for synonymous mutations that don't affect the protein and thus are selectively more or less neutral. N, non-synonymous mutations do change the protein and thus can be selected on. The D makes them both rates. Elevated DNDS often indicates relaxed selection. It can also indicate directional or positive selection, but often relaxed selection. So the first thing we did, calculate a genome-wide DNDS from 1,000 single copy orthogroups representing about a third of the trichromacezola genome. Orthogroup, if you're not familiar, you can just replace it in your head with gene. It just means that if there's a multi-copy gene, we're actually comparing the same copy in each genome, orthologous genes. And so what we find, the same phylogenetic tree, but with all the lengths made even, the trichromacezola highlighted in gray. DNDS is elevated if it's purple, and very clearly elevated DNDS in the trichromacezola clade. This doesn't tell us for sure what's going on, and it doesn't tell us if the signal is really genome-wide or if it's just a few genes driving the signal, but first pass looks like something interesting is going on, might be relaxed selection. So next step, we use a program called HiFi-Relax to look at relaxed selection on individual genes. So we compare the strength of selection on each of about 3,000 individual genes, compare the strength of selection in trichromacezola to the strength of selection in the free living, and this represents about 85% of the genome. And what we find with a corrected p-value of 0.1 is that about 2/3 of the genome is under relaxed selection, whereas only about 2% is under actually more intense selection in trichromacezola. So relaxed selection is what's driving the signal, and it is really genome-wide. Again, linking back to our two ideas of where relaxed selection can come from, might suggest that it's really these bottlenecks, et cetera, that are causing just general drift to happen in the genome. Did I just hit a button? I did not. Okay, last figure I'm going to show. We're going to then curious, is relaxed selection biased towards certain cellular functions? So here, each point in this figure is a cellular function as defined by the KEGG database. The horizontal is how many genes we analyzed in that function. The vertical is how many of those were under relaxed selection. So more relaxed selection, higher up. And if relaxed selection were totally random, then we'd expect everything to be around that genome-wide percentage, that middle 66.5% dotted line. But in fact, it's not random. Some pathways are significantly under more or less relaxed selection. Those are the p-value less than 0.1 are colored. Some others are labeled just because they're interesting, but the three to focus on are those in bold with a p-value less than 0.001. And on top, biosynthesis of secondary metabolites, more relaxed selection. This makes sense in a host environment. You maybe don't need as many pigments. You don't need as many defense compounds. And the bottom, the ribosomal proteins and proteins involved in the light reactions of photosynthesis, they're under less selective pressure, or they're under less relaxed selection, meaning that they're maybe still important. Ribosome kind of makes intuitive sense. It's important for everything. Photosynthesis is really interesting given that we're not sure the degree of integration in the system, and this seems redundant with the chloroplast. Also, both of these functions are among the last functions that stay functional. I use the same word too many times in other endosymbioses. So in conclusion, selection is relaxed across the trichromacezola genome and affects all cellular functions, suggesting that relaxation is largely a result of those bottlenecks, et cetera. Proteins involved in the light reactions of photosynthesis and ribosomal proteins are least affected. And these are also some of the most functional functions in other endosymbionts. This supports the hypothesis that trichromacezola is on a convergent evolutionary trajectory to those of the other intracellular endosymbionts. And with that, I wasn't sure if that comment was them or me. I think it was me. But either way, thank you very much. Thank you to NASA for funding this.
Okay, so thank you for coming. So I'm going to talk about the origin of new fungal pathogens through hybridization. And I'll start with a true story, which is also a sad story. There's this woman who is 80 plus years old. She's pretty healthy despite of her age. She's still traveling around the world, but she wears a heart pacemaker. And the cardiologist detects some malfunctioning of this pacemaker, and he sends her to the hospital for replacement of the device. She goes to undergo surgery. Everything goes fine. But 2 days after the surgery, she starts reporting difficulty in breathing, and the doctors start suspecting an infection. They start taking samples and trying to see what's the cause of this infection, but everything turns out negative. The patient is treated with broad-spectrum antibiotics, And after a week, the things are really worsening, and some organs start failing, and they keep looking for something, and someone noticed there is some yeast-like colonies in some of the cultures. And so they start suspecting for a fungal infection. They treat with antifungals to the patient. They try to identify the species that is causing the infection, but everything is a non-candida, non-albicans candida species, but there is no clear identification of the species. The closest species is Candida inconspicua, but it's not truly that. Unfortunately, the patient does not respond to the treatment and dies a few days later. And the story would have ended here if not because of the doctor was really interested in knowing what was the source of this infection. And he starts looking out for things about Candida inconspicua, which was the closest hit of this pathogen. So he finds out one paper from my lab in which we sequence the genome of that species. It's a rare species that is not very common in infections, but is sometimes found. And in this paper we report is a hybrid species genome. So he sends us his isolate, and we sequence, and we determine it's not quite the same species. It's actually a different species. It's a new species that has never been described. And we actually also find that this new species that is found in this patient is also a hybrid. Interestingly, this species belongs to one clade, which is called the Pichia cactophila clade, which is a clade of yeast that naturally lives associated to plants, and more specifically to cacti. But interestingly, the 3 species in these clades that have been reported to infect humans, including the new ones, they are hybrid species. So I think that's quite puzzling. So I think that story illustrates very well what is going on with the fungi. So first of all, fungal pathogens are neglected, even in the hospitals. They're never suspected unless everything else fails. They are opportunistic pathogens. They exploit the weakening of the host, like in this person undergoing surgery, people with therapy, chemotherapy, immunosuppressed, and so on. Therapy fails many times. We have an issue with drug resistance. Fungal taxonomy is a mess. I told you it was a non-albicans candida species, but actually belongs to a Pichia genus. And it's difficult to diagnose. Actually, in this case, only the whole genome sequencing could diagnose specifically the source of the infection. And I will add a couple of 2 things. So these rare species, like this one, how many they are? I think they are totally underestimated because normally in the clinics, things will stop there. It's a non-albicans candida species, who knows? So it will be interesting to know how common they are really. And also, what's the role of hybridization? In this clade, 3 of the species that normally live in plants, the 3 hybrids can infect humans. Is there something going on here? So I will tell you the story where we first stumbled upon these Candida hybrids, and it was when we were, this is the tree of Saccharomycotina. It's very diverse, and you will see Candida species are all around the trees. So the red dots indicate where pathogens have appeared, and we were interested in the question of how pathogenesis have emerged. No? So we were comparing genomes from pathogenic, non-pathogenic species, trying to find differences. And there was this clade, the Candida parapsilosis clade, that was very interesting to us because there is a gradient in virulence. So Candida parapsilosis is really common, it's the second most common Candida species, and then orthopsilosis and metapsilosis, they are less and less common in infections and less virulent. So we went on to sequence those genomes, and this is where we first started to have problems. So we could not assemble those genomes. They were really hard to assemble. There are these genomes, not very strong, but we found they were highly heterozygous. We were suspecting we had a contamination in the cultures until we were sure we had only, we were looking at a genome from a single cell. And then we had to conclude we were dealing with a hybrid species. Actually, we needed to develop one tool, which is called redundance. If you have a highly heterozygous genome, try it because it helped us to assemble these genomes. Once we assemble the genomes and we remap the reads onto the genomes, we realize these genomes are highly heterozygous, as you will expect for a hybrid. The 2 parental species are about 5% divergence at the nucleotide level, so quite divergent. And we also notice that they have these blocks of loss of heterozygosity, where the genomes have recombined and one copy of the one parental has copied over the other one. And these blocks allow us to recognize different clades of hybrids, because if they are from the same clade, they have a similar origin, and they share some of these LOH blocks, exactly the same boundaries. So we have been investigating this gist over the years, and we have sequenced many genomes. And we have found that the two species, Orthopsilosis and Metapsilosis, so the two rare species are hybrids from different parentals. These parentals have hybridized more than once, so recursively they have formed different clades of hybrids. And interestingly, in the clinics we have found a majority of hybrids. So in Metapsilosis, we never find the parental species. In Orthopsilosis, we mostly find the hybrid species, and only once, I mean, in few isolates, less than 5%, we find the parentals, and only one of the two parentals. So we were thinking, okay, maybe this hybrid has an advantage to spread over human environments and infect people. I mean, I didn't tell you, but these isolates are from all over the world, so they have successfully spread all over the world. So we're wondering where these parental species were. We're thinking probably they are not associated to humans, or they are not able to cause infections, because we never find them in the clinics. So we started to look for them, and the way we did this is to look in the literature to see people identifying these or similar species from diverse environments associated to plants or to other environments. And for this species here, we found several studies that identified these species in marine environments. So we were not expecting this for yeast, but they were quite commonly identified in some marine environments from some warm areas in the world. So we were really intrigued for this, and we were wondering whether, that's a place where the hybrids may be formed or where the parentals live. So we contacted authors, for instance, from this study that is in the Gulf of Qatar. They sample coastal environments, and these are really warm waters. They can reach over 40 degrees in the surface. So we got the isolates, and we sequenced some of these isolates. And this is a picture we had. This is for Orthopsilosis. As I said, we have at least 4 different hybrid clades, and we have one of the parental species, which we call parental A, which is rarely but sometimes identified in the clinics. So when we overlay the ones from these environmental isolates from Qatar, we found that they belong to at least 3 different hybrid clades. So the 3 hybrid clades are there in the environment, and also they are intermingled with the clinical strains, suggesting there is a transmission from the environment and to the clinic. So the clinical isolates do not form an isolated clade. Interestingly, we also found 2 isolates that were fully homozygous, and they were actually very distinct from parental A, suggesting that we actually fish parental B. So now we have the complete picture of the 2 parental species and the hybrids they form. Since we had the isolates, we also looked at the phenotypes, and we could see that this new parental B that lives in the Gulf of Qatar, is actually highly adapted to high temperatures, and the hybrids are somehow intermediate between these features of the 2 parents. We also found that this parent B was also intrinsically resistant to one of the drugs used in the clinics, the echinocandins. So that's also a property that may favor their survival in the clinics. We also did virulence assays using a surrogate model of Galleria melonella, and we saw that the hybrids from the clinics, but also from the environment, and these parental species that was isolated from the environment, they actually had already this virulence potential. So they were killing, in this system, they were showing virulence, also much higher than the other parental. So this means that some features that are useful in virulence are already present in this marine environment. We don't know if they use them there or for what. So the idea is that somehow these hybrids form in this type of environments, and somehow we still don't understand how they break the barrier, and they went to infect humans, and this opens many questions, like what's the role of human activities in mixing different species that then can hybridize and become pathogens to us, and what are the routes they follow to go from this marine environment to hospitals. So this would like an isolated story, but it's not. Actually, by sequencing many of these rare Candida species, we have identified many other cases of rare Candida pathogens that are actually hybrids. About 1/3 of those that are rare are hybrids. And not only rare species, but also Candida albicans, which is the main Candida pathogens. If you look carefully into the genome, you find features that point that this species is actually the long-term descendant of an ancient hybridization. It still has blocks of heterozygosity that you can only explain if there was an hybridization past in time. So what we think is that maybe what we are seeing now in this Candida orthopsilosis species from the Gulf of Qatar is maybe the first steps that happened in the past with Candida albicans. And with that, I'd like to finish just mentioning that hybridization is very common in yeast, that it may have some relationship with the emergence of new pathogens. And I'd like to thank also all the people in my group that help in these projects. So I'm happy to answer questions. Thanks.
So my name is Filip. I'm a PhD student at Stockholm University, and I'm associated with the Swedish Museum for Natural History and the Museum for Naturkunde in Berlin. And today I'll talk about hybridization in birds of paradise. So first here, I was just going to show you the crazy diversity in plumage that is in the group birds of paradise. And it's, for you who don't know, the birds of paradise, they're endemic to New Guinea and surrounding islands, and there are roughly 45 different species, but a lot of them have this like crazy morphological differences. And it's a bit, and they're also known for having these like crazy elaborate courtship rituals, and a lot of the species reproduce through lecking. And despite these like very strong signals of sexual selection, there are a lot of hybrids known between birds of paradise morphologically from museum collections. And just to show how spread this is across the group, so here is like a multicultural phylogeny of birds of paradise, and on each black globe is a intergeneric hybrid between different species that have been identified by morphology. So there is a lot of hybridization going on that has been identified in museum collections because of these like large morphological differences. But then I was interested in if we missed on a lot of hybrids from species that have smaller morphological differences between them. So that is what I wanted to test. So I focused on two groups. This one, Astraka, which are these 5 species, but 2 of them overlap, and Pardigalla, which historically these ones have been placed in the same genre, but now they've been split for some time. But they have like some overlapping distributions, which makes it interesting to look for contemporary hybrids in them. The data we've used for this project is mostly from museum samples, museum skins, and we have roughly like 5 to 10 individuals per species. So for the species that have like large distributions, we have 10. For the small ones, we only have 5. And because we use museum samples, it's important to take like the necessary precautions when you analyze it. So I've used these like purpose-built pipelines that has been developed by my supervisor and lab mates. And we use reference genome for Leucocorax obiensis, which is a phylogenetic outgroup for all of them. So first, when looking at the phylogeny of these species, we see that on the autosomal phylogeny here on the left, most species, or all species, all individuals sort of sort into species. But on the right, with the mitochondrial tree, we see that we have a lot of mixing up in the top group between Astropomeyeri and Stephania, which were these from the same genre that had overlapping distributions. And when we look at admixture-like analysis, we see that we have some signs of recent hybrids. And also, all of them have been, they're all named based on morphology. So none of them stick out phenotypically. They all look like the conspecific species. So we have one hybrid down here between, or one presumed hybrid here then, between Pardigalla brevicauda and Astrapa spendissima, which have like a sliver of admixture proportion from spendissima. And when we look at this ancestry informative markers for this individual, we see that, so we look at fixed sites in the parental populations, and we look at these fixed sites, what identity they have in the hybrid. And we see that we have a lot of homozygote sites that are fixed for the brevicauda, and then we have some heterozygote sites. So it's an indication that this is a backcross towards brevicauda. And when we look at chromosomal patterns of these AIMs in the hybrid, we see that we have this block-like structure that we talked about last talk as well, where we have then in yellow heterozygote regions that are then retained in the genome from the first hybridization event, which is quite cool. And then here on the sex chromosome, we don't have anything because the sex chromosomes don't generally recombine. But if you remember from this slide I showed earlier, we also had a lot of hybrids up here, which all have been identified as Asopa stephania. And we have both males and females. So there's no at least from this limited data set, there's no sex specific effect if you're a hybrid or not. They're from this region, and if we look closer here, we see in the PCA, all the hybrid individuals are these ones. They kind of fall in the middle, which is what you expect. And these ones are from this overlapping sympatric area of the species, whereas these one outside are regions where only stephania occurs. So it seems to be some gene flow going from the sympatric zone into just stephania, the allopatric parts. And then if we look at hybrid triangle analysis for these individuals, we see, so from expectation here, you would expect F1 hybrids to be at the very top of the triangle. This one, which is very cool, I think, has an 50% heterozygote sites and a hybrid index of roughly 50%. So this would be an F2 hybrid, so a cross between two F1 hybrids, which is kind of crazy that you find in a museum collection from naturally culled samples. And then all these ones in red are these other 5 hybrids that are crossing from the direction, from the initial cross into the stephania population. And since we had a lot of hybrids that are from the same species crossing in the same direction, it was interesting to see if there are any general patterns in these hybrids on where you see integration across the genomes. And initially, I wanted to use this AIMS method, but since it's based on fixed differences, and these are a lot more closely related species, there are a lot less fixed sites, so it's very hard to see a pattern in this. So instead, I opted for making a method where I have a lot larger windows, and I use an admixture-based approach. So I lose a lot of resolution, but I still are able to see large, broad-scale patterns. So how it works is just you divide the chromosomes up in larger windows, you calculate admixture in them, and then I use the median value of the hybrid's admixture proportion and see, so I can then look at patterns of how these differ across the chromosomes. So when we look at that, for the microchromosomes, which are chromosomes larger than 40 megabase pairs, we see that we have, towards the ends, we increase in introgression rate, which we also see in the intermediate chromosomes. But in the microchromosomes, we don't really see any pattern. It's kind of uniform, which is also what you expect from recombination patterns in birds based on zebrafinch. We have a lot more recombination towards the ends of large chromosomes, and then you have uniform recombination rates on the microchromosomes. With that, we find cryptic hybrids in Bird's Paradise, We have like some sort of putative hybrid zone going on where we would need more data to kind of be able to look at dynamics of it, but it seems promising that it could be a hybrid zone between these two species here. And we see that the integration patterns follow the expectations from recombination patterns. So with that, I would like to thank my lab mates, people who have contributed by commenting, and my supervisors. The museums have lent us samples and all funding. And if you're interested, recently I have in the lab group 2 papers out on hybridization in birds of paradise. So one in iScience and one in evolution letters that, if you're more interested, you're welcome to check out. Thank you. I'm happy to take questions because I speeded through that a bit too fast. Yeah?
Have you been able to observe, or has anybody been able to observe in the field how hybrids do in terms of attracting mates or being attracted to mates?
So it's not known. There are some indications of hybrids between these two species in the region, but it's this is like the first genomic or genetic description of them, but it's very little known how they perform, the courtship dances and stuff, because they do differ between the two species. Okay, cool.
Okay, hello everyone. My name is Tori. I'm with the Fierce Lab from Florida International University, and thank you all for being here. Okay, let's get excited. We got transposons, canarabditis, and genome size. So genome size is messy, and transposable elements, or TEs, might explain it. You can see that genome size varies greatly between species and doesn't correlate with complexity or gene number. So for example, like mammals, like humans, you might consider more complex than a worm. However, worms can have much larger genomes than humans. And it's well known in the worm community that their genome size is correlated with their mating system. So in dioecious species, their genome size is typically about 20% larger than their androdioecious relatives. And dioecious means male-female, while androdioecious means hermaphroditic and male. Okay. So what is a TE? What is transposable elements? They are jumping genes. They are mobile genetic DNA. And they're classified by 3 things: mechanism of movement, structure, and sequence. The only 2 that I'll be talking about today is mechanism of movement and structure. So you have class 1 transposons, which are copy and paste, and it's pretty clear how they can contribute to genome size expansion because they're literally, by definition, adding nucleotides to the genome. And then class 2 is cut and paste, where they'll take themselves out, move, and insert. For structure, we have autonomous and non-autonomous. So autonomous are those that have all of the necessary gene machinery to move themselves from one place to another. For class 2, that's usually a transposase. For class 1, that's things like integrase. They have a few more. I don't work with class 1 as directly. And then there's non-autonomous, where they've been mutated, they no longer transpose, potentially. They're much smaller. Okay, so here's kind of where my story begins. So we published a paper with the Fierce Lab in 2023. It's called Adams et al. And we found that genome size changes are driven by duplications, insertions, and gene turnover. So the dioecious male-female species were actually expanding their genome size in comparison to the androdioecious hermaphroditic species. You can kind of see that on this phylogeny here. The red arrows are supposed to indicate the androdioecious species. And so the genome size was correlated with gene content, not repeat content. The only exception to this was mutator elements. Mutator elements are class 2 transposable elements. And so we're left with this question, how is class 2 expanding genome size? And my hypothesis is kind of that mutator is associated with the genome size by aiding in the duplications and insertions of genes. So first, let's just look at how mutator is spread out in Caenorhabditis. This is what we expect to see. It's well known that transposable elements will accumulate in the chromosome arms of worms. And when we count them, we do see the same results as the 2023 paper, that in yellow, the androdioecious species have less than the dioecious species. All of them are below 20,000 elements, while the dioecious species are all above 20,000 elements. However, when you count the autonomous ones, the ones that have transposase, intact transposase genes, you don't really see that pattern. And so there might be a lot more non-autonomous in the dioecious species, but activity, if we're inferring activity from autonomous elements, is similar throughout. So now let's ask, is mutator affecting the genes? I did this by just a bed tools intersect 1 kb region on either side. So we see that for most species, they are inserting near genes, likely affecting them. And there actually might be a pattern between the androdioecious species and dioecious species, because the pink is the hermaphrodites, the blue is the male-female, and you can see that the pink have more than are inserting near genes. And then so this is for all mutator elements, and then this one is for autonomous mutator elements. You can see that the pattern might be even more drastic here. And so now we ask, is mutator, we know it's associated with genes, is it associated with duplications? This is for all transposons, not just mutator. We see that it is not associated with duplications, it's rather deletions. So pink is deletion, blue is duplication, dark blue is inversion. So there's a ton of deletions. How big are they? They're actually a lot smaller than the inversions and duplications. The circles are deletions, the squares and triangles are inversions and duplications. While there are a ton of deletions and they're a lot smaller in size, they do result in almost 10 times more affected bases than duplications. So you have 10 times more bases deleted than you have being added. So that was for all TEs. But what about mutator? Mutator is a class 2, so I just broke it into class 1 and class 2 to begin with. We do see that class 2 is very obviously associated with the deletions. And break it down further to mutator. So these are all class 2 elements. So we have mutator, harbinger, hat, and TC1 mariner, and another one. This is less clear, but it does seem like mutator is again greater in the deletions. So it's quite confusing and not what I thought at first. We have mutator largely associated with genes, but it's actually resulting in deletions. It's not helping with the genome size expansion at all. So the question remains open. What's causing genome size variation between the androdioecious and dioecious species in Caenorhabditis? And with that, I would like to thank my entire lab. They keep me sane and smiling and help and support all the time, and especially Jana and Jason for always being amazing mentors, and then funding NSF. So thank you, guys. Oh, I meant to mention that, I forgot. So I just did structural variant analysis, created a VCF file by aligning sequences to a reference. So I can, from that VCF, call deletions, insertions, inversions. And then I intersected that with a transpose on GFF file.
Just a following question, but did you use like software like Lumpy, Manta, and all this stuff to code your sticker elements?
Yes, so softwares I used include like all the repeat softwares, EDTA, Repeat Modeler, Transpose on Ultimate. There's a lot more I'm not mentioning. For structural variants, what was it? We aligned with NGMLR, and I forget the variant caller I used. Yeah. Any other questions? Awesome. Thank you guys so much.